Muscles are contractile tissues in animals that allow for locomotion of the organism itself or of its internal organs. There are many diseases and disorders linked with the muscle, including myopathy and muscular dystrophy. Muscular dystrophy is a group of inherited disorders that involve muscle weakness and a loss of muscle tissue, which get worse over time. There are many different forms of muscular dystrophy and myopathy, but as shown in the chart, the most prevalent is Duchenne muscular dystrophy. According to the Center for Disease Control, an estimated 1 of every 5,000 to 7,000 males aged 5 to 24 has Duchenne muscular dystrophy. Myopathy is a muscular disease in which the muscle fibers do not function properly, resulting in muscular weakness. Certain myopathic states are currently being studied in relation to muscle regeneration, or the regrowth of lost or damaged muscle. This muscle regeneration is closely linked with satellite cells, which are represented with the red arrows, and satellite cells are able to differentiate and fuse to augment existing muscle fibers to form new fibers. Marfan syndrome, or MFS, is a myopathic disorder of the connective tissue with an estimated prevalence of 1 in every 5,000 to 10,000 individuals. Mice were used in this study, and the MDX mice represent the myopathic state of Marfan syndrome in the mice. Fibrosis is the formation of excess tissue and represents abnormal muscle repair. It is seen that there are much higher levels of fibrosis in the MDX mice than in the wild type, suggesting a much less stable muscular system in MDX mice. The study reveals that abnormal regeneration of muscle is apparent with myopathic diseases such as Marfan syndrome. One of the major genetic causes for muscular dystrophy suggested by researchers is the absence of the dystrophin glycoprotein complex, or DGC. The DGC is a cellular complex involved in especially the cardiac and skeletal muscle membrane. It stabilizes and signals interactions and connections between the cytoskeleton, membrane, and extracellular matrix in muscle cells. The DGC is composed of many proteins including dystroglycan, the sarcoglycans, and sarcospan, which all facilitate this connection. Dystroglycan null mice represented by MCK-DG were looked at in the study. Serum creatine kinase was used to identify the amount of muscle damage in the mice. The elevation of this enzyme indicates damage to the muscles. As seen in the graph, the levels of the enzyme are higher in the dystroglycan null mice than in the wild-type mice, suggesting that the absence of this protein leads to muscular damage. The article also notes the observations of regeneration of muscle in these mice. It is interesting to note that the muscular regenerative properties of the dystroglycan null mice were similar to those of the wild-type mice, and that they both retain these properties despite muscular damage. However, in other mutants of the DGC, such as sarcoglycan, this observation was not present. The sarcoglycan gene has been subdivided into various subgenes. Mutations of many of these subgenes, especially beta-sarcoglycan, have been found to induce specific forms of muscular dystrophy, especially limb-girdle muscular dystrophy and Duchenne muscular dystrophy. Meanwhile, mutations in the delta-sarcoglycan gene, as well as the beta-sarcoglycan gene, have also been found to induce limb-girdle muscular dystrophy, along with dilated cardiomyopathy. Cardiomyopathy is the deterioration of the function of the myocardium, or the heart muscle, which usually leads to heart failure. Many drugs are used today in order to alleviate the effects of cardiomyopathy and heart weakening. In this study done by Dr. Kahn in 2001, the L-type calcium channel blocker verapamil is used. This SGC beta null mice represents a beta-sarcoglycan mutant, which shows signs of cardiomyopathy. Using CTN1, or cardiac troponin 1, which is a marker of myocardial damage, it is shown that beta-sarcoglycan null mice treated with verapamil experience less myocardial damage as compared to untreated SGC beta null mice. This suggests the effectiveness of the drug verapamil in order to alleviate the effects of cardiomyopathy in beta-sarcoglycan null mice. The purpose of conducting such research is to better understand and evaluate the mechanisms and effects of muscle disorders such as muscular dystrophy and cardiomyopathy. This may possibly in turn lead to better treatments and cures for such diseases, for as of now only various treatments exist for muscular dystrophy. A cure has not yet been found for this disease. Various protocols and equipment are used in labs in order to effectively study muscle disorders. Northern blots are conducted in order to study gene expression by detection of RNA or mRNA. They are used to observe cellular control over structure and function by determining the particular gene expression levels during differentiation, morphogenesis or development of shape, and abnormal or disease conditions. First, the RNA is extracted from a tissue sample or from cells. The RNA samples are then separated by gel electrophoresis. Since the gels are fragile and probes are unable to enter either, the RNA samples are transferred onto a nylon membrane through a capillary blotting system. The RNA is then fixed to the membrane using UV or heat. Northern blots use hybridization probes, or fragments of DNA or RNA that detect the presence of nucleotide sequences that are complementary to the sequence in the probe. These probes are often labeled with radioactive isotopes or chemiluminescence, which emits light. Bound RNA are then exposed onto an X-ray film for visualization and evaluation. H&E staining is a popular staining method used in histology and medical diagnosis. The use of the stain hemolum stains the nuclei of cells blue. Then using a counterstain of eosin, structures composed of intracellular or extracellular proteins are stained various shades of red, pink, and orange. H&E staining has many applications, but may also be used in the staining of the muscle and its components. In studies of the muscle and muscle disorders, vertebrates such as mice are often used. Human subjects are used occasionally as well, but many of the genes in mice relating to the GGC and muscular dystrophy are homologous to those of humans. Studies done in labs relating to muscular dystrophy often involve the use of mice. Although the mechanisms by which muscular dystrophy and muscle disorders occur in humans are very complicated, simpler homologous genes are available in Drosophila and C. elegans. In C. elegans, the SGN1 gene is homologous with the sarcoglycan gene found in humans and in mice. A potential strain is available for order from the CGC. Satellite cells have been studied in Drosophila in recent years. AMP stem cells in Drosophila can be studied with GFP tagging. In conclusion, there are many different forms of muscular dystrophy, as well as muscle disorders that thousands of people suffer from every day. Scientists must evaluate the mechanisms by which these disorders and dystrophies occur in order to obtain more effective treatments and cures. Here's my bibliography, and that concludes my presentation. Thank you.